Now that we know a little bit about chemistry, the atoms, the periodic table, and how atoms come together to form molecules, we already looked at a very important molecule, which is water. We're going to move on now to see some of the other really important compounds that make up living organisms. They're kind of separated into two groups, not kind of, but they are. The inorganic compounds versus the organic compounds. Inorganic compounds are very important. We think organic, but inorganic are things like water, oxygen, carbon dioxide, and the inorganic salts. No living organism can make it without these inorganic substances. We'll talk more about those later, but I think all of us have a general idea of why water and oxygen, carbon dioxide, and these inorganic salts are so important to us. Chapters 3 and 4 are what we're looking at this week, and they hit mostly on the organic compounds, but I just like to throw the inorganic compounds in there to let you know that it's not just about the organics. With that said, let's go ahead and move into our organic compounds. Basically, the definition for that says that anything that contains carbon. But if you looked at the inorganic compounds, you saw that CO2 contains carbon. It's an exception because it's a gas. And like all things, there are a few exceptions, but for the most part, if it contains carbon, it's organic. And we're interested in looking at these things and how they act and behave in living systems. It's called biochemistry, and we'll be looking at more and more of that as we move on. We're just familiarizing ourselves with these molecules at this point, but at some point we're going to be looking at the physiology, how these things are actually working. When we talk about our organic compounds, there are 4 main organic compounds: carbohydrates, lipids, proteins, and nucleic acids. The first thing I want you to do is look at this little box that I have here of the organic compounds. They're called carbon compounds here, but you'll see the titles: carbs, lipids, nucleic acids, proteins. And I really want you to get to know this chart. These are things that at some point you're just going to have to know off the top of your head when we're talking about these things. Carbohydrates, what are they? Well, they're sugars and starches. So if we're looking at our bread and potatoes, lima beans, those are heavy starches. Those are carbohydrates. But so are the sugars. And by sugar, I don't mean refined sugar like white and brown sugars, things like that. I mean fruits and vegetables. All forms of sugar are carbohydrates. The lipids are the fats, the oils, and to that we can also add waxes. Fats, oils, and waxes are lipids. Nucleic acids are called nucleotides. What I mean by that is that this molecule here, this nucleotide, as we put those together, we start to make nucleic acids. And nucleic acids are very important because they're our DNA and RNA. We'll talk about that more later. Proteins, their building block is an amino acid. As we put amino acids together, we start to create proteins. So carbs, lipids, nucleic acids, proteins, they're made up of sugars and starches, fats, oils, and waxes, nucleotides, and amino acids. And then we want to break that down even further and look at what elements make up these different organic compounds. And you'll notice that carbon, hydrogen, and oxygen are in every single one of them. In fact, carbs and lipids are made up only of carbon, hydrogen, and oxygen. That's all they're made up of. So how do we tell the difference? Well, next week when we start to look at carbs and lipids, we'll see how to do that. Next one, if you look at, I kind of like to jump over to the proteins because there's just one other element added, and that's nitrogen. So things with nitrogen are proteins. Proteins, carbs, hydrogen, oxygen, and nitrogen. So look at nucleic acids, we add yet one more, phosphorus. We have, just like a protein, carbon, hydrogen, oxygen, nitrogen, but we also add phosphorus to make a nucleic acid. Everything on this box is going to become something you need to No. So try and memorize that. Now, looking at chapter 3, we're talking about proteins, and proteins make up every single structure in your body, and every single function of your body has something to do with a protein. Incredibly important. They're a structural material, an energy source. They make hormones, neurotransmitters, receptors, enzymes. They're immune antibodies, and their building blocks are amino acids. But when we look at these things, hormones, that's how a living system communicates to different glands in the body. Neurotransmitters, that's how the nervous system communicates. Receptors, that's how muscles communicate with the nervous system. Enzymes, they're the catalysts for everything that happens. Immune antibodies, that's talking about the immune system. So when you look at that, you can see just how vital the protein is. When we talked about proteins, we said that the building block of protein is an amino acid. Amino acids form very small proteins called peptides, and then actual proteins are formed from peptides, and they're much larger. Amino acids are called monomers. They're called monomers because it's a single structure to make a protein. Mono, one, monomer. Amino acids are the monomers. They build the peptides and proteins. Proteins have what's called an R side group, and that side group is going to determine the characteristics of each of those amino acids. We'll talk a lot more about that in a few minutes. Peptides are polymers. Now, unlike a monomer, polymer is made up of lots of those amino acids. They're made up of 2 to 100 of them, and they're linked by peptide bonds and are linear chains. Again, we'll look more at that later. This is more just to introduce you. 2 amino acids are called a dipeptide, di meaning 2, 3 tripeptide, tri meaning 3, 4 is a tetrapeptide. Oligopeptides are 2 to 20 amino acids. Peptides are 2 to 100 amino acids. And at this level in introductory biology, I don't expect you to know that or its relevance, but I'm just showing you that to show how we can put these amino acids together to form a lot of different types of protein. Peptides allow effective communication by carrying biochemical messages from one place to another in the body, and that would be things like our hormones. Proteins are made up of folded polypeptides. When we talk about proteins, we can't get away from the structure of proteins. The structure of the protein is going to determine what it does. And when you look at what proteins do, all of the different functions that they have, there's all kinds of different ways that they're going to fold and behave. Sequence of amino acids determines the characteristics. It alters a single amino acid, can cause a gene mutation, which most often will result in a non-functioning protein. Stable three-dimensional structures, the hierarchy of 4 levels: primary, secondary, tertiary, and quaternary. Now we'll talk a lot about these different structures of the proteins as we move on. There are tens of thousands of distinct proteins in our body, and they're made up of amino acids. You have to kind of wrap your head around the fact that we only have 20 different amino acids. But these 20 different amino acids are going to make thousands and thousands and thousands of different proteins. And I guess you can kind of equate it to like the alphabet versus how many words are in even just the English dictionary. We have, 26 words in the alphabet? And if you look at a good dictionary, it might be 4 inches high, and every page has hundreds of words on it that are tiny, and the front and back of every page has words on it. So to even try and count that number of words is kind of a waste of time, I would think. But the point that I'm trying to get across is it's made out of 26 letters. 26 letters makes all these different words. So you can imagine how many proteins only 20 amino acids have the ability to make. So when I say we only have 20 different ones, don't think for a minute that there's not a lot of different proteins, because there are just as many different proteins as there are words that we can make out of the English alphabet. Each have a basic amino acid structure around the central carbon. It's called the alpha carbon. If you look over here at the amino acid structure, this is the basic structure right here, and you're always going to see it. Every one of those 20 amino acids has this structure. We have an amine group to the left. We have a hydrogen in the center, and we have the carboxyl group to the right. This is our central carbon right here. It's our alpha carbon. From the alpha carbon, we get all of these different molecules attached to it, those valence electrons. Now notice this one down here, it's called the R group. Whatever attaches here to the alpha carbon will determine the amino acid, because they all have this basic amino acid structure. So remember that we have an alpha carbon attached to an amine group, a hydrogen, and a carboxyl. It's the R group that's going to determine our amino acid. If we look here, these are the 20 amino acids. And if you look at every one of them, you see that basic amine group on top of each one of them is the basic amine group. It never changes. What does change is the R group, whatever is attached here to the alpha carbon. They're all different. And because they're all different, that's what gives each of these amino acids their own special qualities. An amino acid gets its name from two primary functional groups, the amine group and the carboxyl group. Here's our amine group again, our hydrogen, our carboxyl group is over here, and here's our R group. The amine group is called amino, and this group right here is actually an acid, amino acid. That's how we get the name amino acid, is from these structures. The carboxyl group is acidic, the amine group is a base. Now we talked about acids and bases. So one side of the alpha carbon is going to be acidic, this group, and one side is going to be a base, this group. Amino acids are neutral until we consider the R group, which gives the amino acid a positive or negative charge. Now, we have lots of things going on here. We have the carboxyl group that is an acid, and it's got a negative charge. We have the amine group that is a base and has a positive charge. So if I have one acid and one base, I'm basically neutral, right? And if I have a positive and a negative, I'm neutral. So this amine group right here, this amino acid, is neutral. What will change that is whatever happens here at the R. If I put an R group on here that's more acidic, then I have an acid. If it's more basic, I have a base. If it has a positive charge, I have something positively charged. If it's a negative charge, I have something negatively charged. But until I add that R group, my amino acid is always neutral. Any questions, make sure you get a hold of me. All right, so now we have to look back at some of the terms we learned in our last chapter. Polar. What does polar mean? Well, polar means it's more positive on one side and negative on the other. Those charges are, it kind of works like a magnet. And a hydrophilic, remember hydrophilic? It likes water. So we have polar hydrophilic amino acids. They have a charge and they're R group side chains. So these, you can see, come along here, and they're charged. They interact readily with water or other charged molecules. The polypeptide R group chains twist to interact. The hydrophobic groups twist away from the side chains, forming protein folds. Serine, the hydroxyl group, is polar. Serine, it has another polar group right here. And here we have another polar group. Okay, so what is this saying? You don't have to memorize all this. What I want you to do is try to understand it. you do have to know that we have polar hydrophilic amino acids and what that means. What does it mean? It means it's charged, unevenly charged, and it likes water. Why is this important? It's important to understand these next couple of slides because that tells us how it's going to behave and how it's going to twist and turn and fold. Remember, I told you we have all these proteins from 20 amino acids, and they all behave differently. Why? Well, this is why. Because if it's polar and hydrophilic, it's charged, it's going to be attracted to water, and it's going to face water, right? It's going to want to bond with water. Whereas if we look here at the next one that's hydrophobic, that means it doesn't like water. So what's it going to do? It's going to fold away from the water. So now you see how all of these different proteins can try to fold and form all of these different structures. Acidic amino acids, they're a functional group with an OH group. They're acidic. They have a low pH. They accept protons. So these acids are going to accept protons, aren't they? They're going to accept hydrogen. A nonpolar hydrophobic amino acid, what's going to happen when this thing gets put in water? It's going to go as far away from the water as it can. It's hydrophobic. It doesn't like water. It's going to fold against the water. It's going to fold to the inside. Basic amino acids, they contain an NH group capable of adding hydrogen. So these bases are going to be able to pick up hydrogens, and the acidic are going to pick up the carboxyls, and the basic are going to pick up hydrogen, which is going to alter the way that this is going to be folding and behave. Polar versus charged amino acids. Polar molecules, their electrons are not evenly distributed around the outer shell, and when polar molecules encounter nonpolar, they exclude them. They form covalent bonds. And charged molecules are going to form ionic or covalent bonds, and they are considered polar. Why are we going through all this again? It's because it's just explanations about how these different proteins are going to fold and behave. Different structures, whether they're linear or form tubes or just fold into globs. These are all different ways that these proteins are going to behave. If you look at your hair, if you look at your muscles, they're very, very different. And it's because of these different properties that we're going through here. Now let's look at this chart again. We saw it, and all we're interested in the first time is to see our basic amine group and see how the different R groups are what determine which amino acid this is going to become. But if we look again, now we see non-polar side chains, polar side chains, electrically charged side chains. All of these are going to have different behaviors. Understand why you have polar and non-polar amino acid. What makes the variable group hydrophobic or hydrophilic, protonated, deprotonated, acidic, and basic amino acids. And why do we have to understand it? Again, it's just a repeat of what I've been saying. It just tells us how it's going to behave. Now, if we look just briefly for a second at this picture, it's not something you're totally going to understand right now, but you will later when we start talking about the cell membrane. These are proteins that are lodged into the cell membrane, and they become very, very important. Now, they have hydrophobic regions that do not like water, and they like hydrophilic regions that do like water. Now, on the outside of a cell and the inside of a cell is covered in water. It's completely saturated in water. And what keeps these proteins in the membrane is the activity of the hydrophobic and hydrophilic regions. The hydrophilic or phobic regions, if they happen to slip down into that water, they're going to pop right back in here where they're away from the water. Whereas these hydrophilic regions are going to stay right out here in the water where they're nice and happy. And this is a very important concept to understand because this is the basis of the cell membrane. This is what holds things together, is this hydrophilic ability. And this is why it's so important that we don't dehydrate, because if we dehydrate, this whole system falls apart. We'll talk more about that as we move on too. Okay, there's different categories of proteins, essential, non-essential, and something called a conditional non-essential protein. The essential protein, it just means that we can't produce it. We must consume it. And a regular daily supply of these essential building blocks have to be eaten. Our liver can't process it. In other words, it's something we have to actually eat. Whereas the liver can process the non-essential proteins. They're produced by the body. It's not an essential part of your diet, because what the liver will do from the foods that you eat is break them all down to their elements, and then it can put things together like a puzzle. It can put proteins together that we need and things like that. So the non-essential are things that we can actually produce. Essential, we have to consume. Conditional non-essential, if you get sick, or if you have some kind of stress or an injury or illness, these non-essential amino acids become essential, because maybe we can't produce them, and so now we have to start eating them because our body's not working properly. And here, this is just a chart of the essential, the conditionally non-essential and non-essential amino acids right here, just so you can get an idea of what that looks like. And then we have some other charts that you can go through on your own. They talk about the essential amino acids and the actual benefits of those, why we need them. And here again, now we talk about our protein structures. 4 levels of proteins are distinguished from one another by degree of complexity and the polypeptide chain bases on complex shapes that include various folds, loops, and curves. You'll understand what I mean by that as we move on. We have primary, secondary, tertiary, and quaternary. And it starts out as a very simple complex of amino acids, and gets more and more and more complex as it moves on. Shapes cannot be altered or they lose function. Every single one of these bends and turns and folds of a protein are intentional, deliberate. Nothing about it is random. This is what they're supposed to do. This is what the DNA tells them to do, because those are the folds that determine what's called domains, and then it has a function. It has something that it has to do in the body. And if it loses any of that shape, if it loses any of those folds or turns, then it's going to lose function. We don't want that. Basically, it says here, ignore polarity on the carboxyl and amino groups and only look at the side chains. Why would that be? That's because just that basic portion of the amino acid is always going to be neutral. We don't have to worry about it. The side chain is going to determine all of that activity. Let's look at primary first. Primary, it's just linear. It's a linear sequence of amino acids connected by a peptide bond. And peptide bonds are dehydration condensation bonds, where we either remove or put in water. That's why it's called dehydration, because it's an H2O. I'll show you that here in just a second. They're formed by covalent bonds between alpha amine groups and alpha carboxyl groups. So one of the amino acids is going to form a peptide bond to the other amino acid. And I'll tell you what that means right now. Here we have formed by a covalent bond between the alpha amine group, and here's our amine group right here, right? And this is here's an amino acid, here's an amino acid. So the amine group of this amino acid is going to form a peptide bond with the amine or the carboxyl group of this amino acid. Now look what happens when they form their bond. Do you see this OH group right here, this hydroxyl group? And then do you see this hydrogen? Do you see how they came together to form a water molecule and left? This left, that's called dehydration because it leaves. And as soon as that left, if you look over here, do you see now the bond between this amine group and this carboxyl group without the hydroxyl and the hydrogen? You see that? With this gone, we have our carbon here and our nitrogen. Do you see how the carbon and nitrogen are bound together? So that's how one amino acid comes together with another amino acid. This is always going to happen on your amino acid chain because of this group, this top part right here, this main amino acid group, they're always going to bond together. It's going to be the side groups that are kind of dropping down in here. You see what I'm talking about? If I'm confusing, let me know. But here's one amino acid, here's another amino acid. The carboxyl group of this one and the amine group of this one are going to form a peptide bond by removing water. If this hydrogen comes off, this hydroxyl group comes off, we leave an opening here on this carbon, a valence electron, and we have a valence electron here on this nitrogen, and they're going to come together, and we've lost water, dehydration. Now, if I were going to break this apart, it would work the other way around, wouldn't it? I would put the water back in, and that would be called a condensation bond. So we dehydrate and we condensate to either break these amino acids or combine these amino acids or break them apart. And we'll have something that we're going to experiment with in lab this week so that you can see that activity. By doing this, what we're doing is forming this long, straight chain. Do you see if I keep adding them on, adding them on, adding them on, they're just going to form a long, straight chain. And that's what we mean by a linear sequence. And it is a primary protein. It's a very simple protein. Here's some other pictures of that. Here's how we remove our water and blah, blah, blah. And here again, we're doing it. Same thing. Here's our peptide bond that we've formed. Okay. Here's another one. Do you see how we just add an amino acid to another one, to another one? And here these lines are the peptide bonds. So we've removed water. We've been able to bind that carbon to that nitrogen. And we just keep making more and more and more and more of a primary structure, which looks a lot like a necklace. It's just a one line. If you fold it straight out, it's a nice straight line, or you can kind of bump it up a little bit, but it's still a primary structure. A secondary structure is a linear sequence of proteins, but they begin to fold into regular repeating patterns. And this structure comes from the backbone, hydrogen bonding, a covalent bond interaction. So if you look at the backbone, now instead of looking here, we're going to start looking up here. Do you see the hydrogen? Here's where we start bonding to form a secondary protein. These bonds, hydrogen to oxygen, do you see? Now we're looking at, instead of looking at side to side, we're looking at the top and bottom. See the hydrogen and the carbon? And we start to go from one to the next and bond that way, and it starts causing us to either twist into a helix, kind of like this, or into something called the pleated sheet, where we have one that's folded over and then they bond this way. So they'll bond one way or the other, but do you see now how they're bonding? This hydrogen is going to bond to the oxygen. Here, see that? That is how we form a secondary structure. So the primary structure is formed by the amine carboxyl groups, and the secondary structure is formed because now we're bonding, we're having the hydrogen bonding. Two particular types of the alpha and beta. We are looking here. This is the helix shape. Here is the alpha. And our shape here of the pleated sheets is the beta. Here's some more looks at those. Look at those structures. Our alpha forms kind of a coil, see? Helix. And here's the pleated sheets. And there's another look. And this is really nice because you can really see the molecular bonding. A lot of this is just looking at these various figures and trying to understand what it is I'm pointing out to you. See this, when we talk about a secondary structure, do you see how the hydrogen is bonding with the adjacent oxygen and we're forming the secondary structure? The primary, again, when we look at how we form the linear structure, find it. Here it is. Okay, so when we form the linear structure, we're dealing with the side-by-side this way. That's how we make a primary protein. And when we are making a secondary protein, we're using the not side-by-side, but adjacent, kind of up and down. Like here. And that's how we make a secondary. So here we already have primary proteins. You can see that, right? Because we have our linear structure that had the peptide bonds formed by the amine group and the carboxyl group. So we have these primary structures. And now those primary structures are starting to come together because we're forming bonds still on the basic amine group. We're not touching the R groups. Do you see that? None of these touch the R groups. It has nothing to do with it because the R groups start to come in to play now in our tertiary structure. Primary structure, we're forming peptide bonds between the amine group and the carboxyl group. Secondary structure, we're forming bonds between the covalent bonds between the hydrogen and oxygens of the adjacent primary peptides or proteins. And here again, you can see that we're forming these bonds. Again, we're not touching our side chains. Tertiary structure, that's a three-dimensional folding. Now we saw how the primary is nice and linear. We saw how the secondary kind of bonds with other primaries to form either a helix or these pleated sheets. And now we look at tertiary, and it's the first time we start to see kind of a 3D folding. It's a final protein configuration, and this involves the side chains. That is how we form these. And that's where all that acid and base and positive and negative and hydrophobic and hydrophilic stuff comes into play, because now is when it's going to just start wiggling all over the place and folding, 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 until we get something that looks like this. See, here's our secondary protein helix. Here's our secondary protein folded sheet. But now, look what's happening. All of our side chains are starting to come together. And they're going to end, along with those side chains, we have that hydrophobic, hydrophilic. Some of them are going to fold in, some of them are going to fold out, until we start getting proteins that look like this. It looks like a bunch of, where you take ribbon and you curl it. That's what it looks like. Now, we talked about all the different structures. So hopefully by now you understand. Primary structure, peptide bonds between the adjacent, or not adjacent, side-by-side amine groups and carboxyl groups. Secondary, We have formed covalent bonds between the hydrogens and oxygens that put these primary structures together. Tertiary, the side chain groups now start bonding, and they start forming all kinds of twisting and turning and twisting and turning. Our last structure is the quaternary structure. It's a combination of tertiary structures. So basically the quaternaries, when we start putting these tertiary structures, these very complex, folded, turned structures together. So imagine putting 2 or 3 balls together. That's kind of what it would be. It's a combination of tertiary structures. The interaction of the positively charged amino group and negatively charged carboxyl acid groups in the subunits form a quaternary structure. There are 2 categories of proteinaries, globular and fibrous. The globular proteins, globular is sort of spherical or round structures. They're folded and compact, usually spherical. Things that make globular proteins are like insulin, enzymes, hemoglobin, which rides on red blood cells. They're compact and they're ideal to travel through narrow blood vessels. Then we have the fibrous ones. Now the fibrous, if you think of a fiber, it's elongated. It's kind of long and stringy, like collagen, myosin, keratin, fibrinogen, and we'll look at those more here in just a second. This is actually a very short lecture. The shape is critical to its function. It's maintained by types of bonds. Denatured proteins are proteins that have undergone changes in shape because of temperature, pH. All of these things have to be very precise. The environment has to be precise, or proteins will unfold, unbond, and they will lose their function. 4 structures: primary, secondary, tertiary, and quaternary. Now here's the examples of the quaternary. Here's fibrous. Do you see how it's just tubes? It's kind of elongated, and they come together. See, this would be 1, 2, 3 tertiary proteins that came together to form a bigger, stronger unit. Here's a globular protein. This is one that has a very folded structure that is usually quite round. And here again, a fibrous protein. These are examples of fibrous proteins. Connective tissue, fibrous tissue, like ligaments, tendons, skin, cartilage, bone, blood vessels, cornea of the eye, intervertebral discs, muscles, and the gastrointestinal tract. Keratin are things that make like claws, beaks, hair, the cytoskeleton, nails, hooves, feathers, horns, fish scales, tough outer layer of skin. And of course, you know that this is just a small sampling. It's just to give you an idea. But these are what some of the things that the fibrous proteins actually come together to create. The globular proteins, do you see how perfectly round they are? This is an insulin, and here's hemoglobin. Hemoglobin, these things ride around on your red blood cells. And if you look here at this hemi unit, there's 4 little hemi units. And if you see the dot on these hemi units, that's iron. And this is what actually carries blood. This is what carries blood from the lungs to the body, are these little discs of hemi with the iron on them. And that's why they say if you have iron poor blood, you don't have a lot of energy and stuff. And it's because you're not getting enough oxygen. So we always have to have plenty of iron to go on these discs of hemi. And that is why it's called hemoglobin. This is the hemi right here, these discs that carry the iron. And the globulin are these 4 little structures that you see. You see those? Now, what this is 1, 2, 3, 4 different tertiary proteins all stuck together to make this quaternary protein of hemoglobin. Again, there's our levels, and I think we've talked an awful lot about them. Maybe not quite enough on the quaternary, but the quaternary is just tertiaries put together. Okay. And lastly, the thing we're looking at are our nucleic acids. And I'm not going to go into as much detail as I would like to right now about nucleic acids, because we're going to start talking about them in depth later on as far as the nucleotides that create them and what their functions are. Basically, the nucleotides are two things. They're RNA and DNA. If you look at RNA and DNA, you'll see they both have Na, that's nucleic acid, and R is ribonucleic acid, and D is deoxyribonucleic acid. And basically what those are, RNA makes proteins, and we already know how important proteins are, but RNA is what makes those proteins for the body, and we'll talk, like I said, we'll talk a lot more later about that. DNA is our genetic code. Those are our genes. That's what makes us who we are. That's the hereditary information that we got from our parents. And again, that's going to become really important, but later on in the semester, we're going to be looking more at, okay,